Okay, so like I mentioned, our goals for today are to talk about the cardiovascular system in the applied fluid medium, that being blood, right? So last time we left off, we had discussed each of the components of whole blood. So there's two major parts, plasma and formed elements. We have a layer called a buffy coat that precipitates out in a centrifuge that's part of the formed elements, but it's specifically the platelets and the white blood cells that are a little bit less dense than all of the erythrocytes that make up the red blood cells. All right. So if we talk a little bit about some of the dissolved components of plasma, some of the important proteins and ions that we find in there. So we have albumins, which can help maintain osmotic pressure. We use them to make sure that we don't lose water to our surroundings. It's kind of the mammalian adaptation for retaining water effectively inside of our body. Other organisms do this a little bit differently. For example, some saltwater fish, instead of having things like albumins floating around, they actually use their own nitrogenous waste in their bloodstream. So they have their own urine and even sometimes some of their fecal matter that's floating around in their bloodstream that they use to make sure that they don't lose water to the surrounding saltwater environment. Let's get Chris back in here. So we use albumins to help kind of maintain our osmotic potential, and then we use them to also destroy some foreign substances and can operate as part of the complement system for assistance with antibody function. We can use globulins as well for transport molecules to house some of our our fat-soluble lipids. Lipids are fat-soluble. Our fat-soluble hormones is what I'm trying to say. So those can be floating around and be attached to globulins. And then we use fibrinogen, which is again a fat-soluble protein that can be floating around, and it helps assist with the formation of clots. So we'll discuss those today. We have a bunch of different kinds of trace ions that can be floating around, and even ones that aren't trace ions like sodium and chlorine. Those can be floating around in the bloodstream at any given point in time. We have hydrogen ions, hydroxide ions, and things like bicarbonate ions, which are a direct correlation to the chemical environment of our blood. So if it's too acidic or too alkaline, we can modify the concentrations of those ions in our blood with the help of a nice little handy enzyme called carbonate anhydrase. And so we use that to help ensure that our pH is relatively stable. And then, most kinds of nutrients, amino acids, lipids, carbohydrates, all of those things can be floating around in the blood as well. And then dissolved gases. So when we talk about hemoglobin here, hemoglobin's main job is to bind oxygen. Now, the way that works is kind of interesting, but most of the time, it'll only be bound to oxygen, and then it can modulate how it binds to that. It will bind to carbon dioxide if it needs to, but most of the time carbon dioxide will end up getting dissolved in our bloodstream, so in the plasma. But we can use hemoglobin in a pinch to bind some of that carbon dioxide too. And then other kinds of waste products. So we do have some nitrogenous waste that can make it into our bloodstream. Usually this can be in the form of something like biliverdin, which is a kind of waste product made of breaking down red blood cells, as we'll see in a little bit, before it gets converted into bilirubin. And then we have, like I said, other kinds of regulatory substances, mainly humoral elements like natural killer cells and other kinds of hormones that can, send signals throughout the bloodstream. All right. So then, if we take a look at each of the formed elements of our blood, so we've got our erythrocytes, red blood cells. One of the main interesting features about them is their shape. First of all, they're this biconcave disc. So this outer ring is a thicker portion of the cell, and then this inner flat part is flattened, it's squished, it's thinner than the outer ring. So it's called biconcave for that reason. The other unique feature about these things is that they don't have nuclei in them. So red blood cells have no nucleus, they have no ribosomes, so they can't manufacture proteins, and they don't have any mitochondria, which is interesting too, because then they can't undergo aerobic metabolism. So red blood cells from the start, when they're developed through stem cells, kind of have a set lifetime before they end up breaking down and getting worn out. So, it's kind of interesting how we develop them because other animals have red blood cells that contain nuclei in them. A really great example of that is frogs. Frogs have red blood cells with nuclei in them, so they don't have the same kind of erythropoietic system that we do. All right, and then we've got our leukocytes, or some of the other formed elements here. Leukocytes can be typically broken up into two categories. We have granulocytes and then agranulocytes. The granulocytes are called that because they have these granular components in their cytoplasm that get stained and show up under a light microscope. So neutrophils are interesting. They're one of the most common leukocytes that we have in our body. So it's got a nucleus with either 3 to 4 lobes connected to one another via filaments. We use these to phagocytize microorganisms and other substances. So they're one of the kind of main initial responders to an infection. So if we have a laceration and we get a cut or something like that, one of the main cells that gets to that site first and is part of that initial inflammatory response is a neutrophil. So these guys will then go and swallow and metabolize and break down any kinds of bacteria or other pathogens that are trying to invade us. Then we have basophils, so nuclei with 2 distinct lobes. Cytoplasmic granules will stain bluish purple and roughly 10 to 12 microns in diameter. These guys are going to initiate the inflammatory response. So these are part of our complement system. So we'll talk about this more once we get into our lymphatic system, which I think is chapter 14 for us. I think it is chapter 14 is when we get into lymphatics. So we'll talk about this kind of sequence of proteins that we use in order to kind of trigger a humoral response for pathogen destruction, right? So basophils will go into the site of an infection or the site of an injury, and they'll release histamine, which promotes inflammation. So inflammation responses they carry with them a whole host of other things that go on. One of them is dilation of blood vessels and increasing of the pores inside the blood vessels. Blood vessels have these things in them called fenestrations, which allow for things like neutrophils and other kinds of macrophages to enter the site of an injury or the inflamed site and leave the blood cell and then start fighting off pathogens and bacteria in the interstitial space. So that's kind of cool. Part of the problem with too much histamine is that it can drop blood pressure really, really quickly. So if you go into something like anaphylactic shock, for example, or if you have a severe allergy to some kind of food or medication or something like that, usually what injures people the most from something like anaphylactic shock is the dramatic drop in blood pressure that's pretty much that occurs all over the body. It's not just a localized area. So if blood pressure across the entire body drops, then that messes with things like partial pressure gradients for gas exchange between the bloodstream and organs. And it's basically bad news bears because then the organs aren't able to get their oxygen that they need. And they also can't dump off the carbon dioxide that they need to. So maintaining blood pressure is very important throughout our body. Things like basophils will release histamine that can decrease blood pressure, but they will generally do so for only short periods of time to trigger an initial inflammatory response and then activate other kinds of neutrophils and things like eosinophils that can go in and attack and fight off specific kinds of parasites or whatever is in that area. So eosinophils tend to be a little bit larger than our neutrophils and basophils here. These guys are around 11 to 14 microns in diameter, and their granules stain a bright orangish red color, which is kind of cool. So these guys also participate in inflammatory responses of allergic reactions and things like asthma, and can attack certain worm parasites that may enter our body as well, usually in our intestines. And then we get to agranulocytes that don't have any cytoplasmic particles that stain different colors. So these guys, lymphocytes, relatively small, 6 to 14 microns in diameter. So these guys produce antibodies and other chemicals responsible for destroying microorganisms, help contribute to allergic reactions, responsible for graft rejection or things like agglutination reactions, where with blood transfusions, if you get the wrong blood type, then you can have an agglutination reaction where the red blood cells get destroyed. So graft rejection, tumor control, and regulation of the immune system, all are regulated by lymphocytes. Then we've got monocytes here, which have this large kind of kidney-shaped nucleus. These are phagocytic cells that kind of circulate around in our bloodstream. These guys are pretty cool because they'll enter the site of an inflammatory response and they'll leave the circulatory system via those fenestrations in the blood vessel, and then they'll transform into macrophages and they can go and destroy bacterial cells. So these are part of our T cells that are, you know, one half of our immune system, and then the lymphocytes are B cells that are part of our immune system. The monocytes, there's like a couple different kinds of them. Some of my favorite ones are what we call cytotoxic T cells. Can't wait to talk to you guys about those. They're so cool. Yeah, it's so awesome. But anyway, so agranulocytes and granulocytes. And then we have these little cell fragments that arise from a specific kind of cell called a megakaryocyte. And so megakaryocytes will end up kind of dissolving as part of their life cycle, and they'll break down into fragments. So these little fragments are what we call platelets, and these platelets are responsible for activating clots, right? Or not activating, but triggering the clotting process, rather. So if you have low platelet count, right, then your odds of clotting due to an injury are lower than somebody who has normal or higher platelet count, right? There's all kinds of things that can affect whether or not the body clots appropriately, and we'll talk about a few of those. So platelets help form plugs at the site of an injury to a blood vessel. They'll release certain chemicals that'll trigger and activate other platelets in the nearby area as part of a apocrine response to, you know, an injury site. And so we trigger this little positive feedback loop that continues to build up and create more platelets to form the plug. And then once that plug is formed, then we can convert all that stuff into a clot via fibrinogen, right? Okay, so here we've got a nice little flow chart. I would recommend, you know, using this as a study guide for understanding the life cycle of different parts of the formed elements of blood. So again, when we talk about hematopoiesis, what we're referring to is the development of blood cells. Whether they are red or white, they all come from the same kind of stem cell called a hemocytoblast, okay? So these hemocytoblasts contain the information to transform into either a erythrocyte or a white blood cell or a leukocyte, okay? So a couple different trajectories here. I won't necessarily go into depth with all of them, but so, you know, we've got two main pathways: lymphoid stem cells or myeloid stem cells, lymphoid stem cells, and then one branch of the myeloid stem cells will develop into mono and lymphoblasts, which will eventually develop into mono and lymphocytes. That's pretty self-explanatory. But the prourethroblast is the branch off of the myeloid stem cell that will end up developing into our mature adult red blood cell here. So it's kind of interesting too, is late in the development, they start out with a nucleus, but part of the directive of the cell is to eject that nucleus, get it out, it gets out, right? What's kind of cool about the megakaryoblast is these are super mega ultra enlarged stem cells for blood, and then they end up kind of lysing and destroying and dissolving themselves, and they create these little fragments that we call platelets, all right? And then we've got these myeloblasts that will develop into progranulocytes, which can specialize into one of our three different granulocyte white blood cells. So basophilic, eosinophilic, and neutrophilic will tell you which ones they'll turn into, right? Okay, any questions before we move on to hemoglobin? Hemoglobin. All right. Okay, so hemoglobin is a pretty cool protein. It's one of the few proteins in our body that actually has what we call a quaternary structure. So we have other proteins that exist in quaternary form, absolutely. But hemoglobin is pretty advanced in that it's a tetrameric quaternary protein. So there's 4 different subunits that belong to this protein in order to make it function correctly. And it's kind of crazy how little has to go wrong in order for the development of this protein to not go the way that it's supposed to. So each of these globin molecules, we have globin alpha 1, globin alpha 2, globin beta 2, and then globin beta 1. Each one of those globin molecules is a protein on itself. But to make hemoglobin, we have to fuse all four of those branches together. And we do that through disulfide bridges, if you guys remember those way back from chapter 3. Within each one of these globin molecules, we have a hydrocarbon chain that's bound in the center to 4 nitrogen atoms. And then at the center of that, we have a iron ion. So it's not really depicted technically correct here. It's in its ionic form, which is a 2+ charge cation. Okay. The significance of that is this iron atom that's in its ionic form will bind to gases like oxygen and carbon dioxide, which is pretty cool. So at any given point in time, a hemoglobin molecule is capable of binding 4 different molecules of breathable oxygen. Now, does that actually happen? No, not necessarily. It's a rare case when your hemoglobin molecule is completely bound to 4 different oxygen molecules. Because what we find is that with hemoglobin, as it continues to bind more and more oxygen, it ends up getting this thing called diminishing marginal returns, which I'm sure we're all familiar with, in terms of candy bar consumption, right? I don't know what your guys' favorite candy bars are. Personally, I am a Kit Kat and Snickers gentleman. I love Snickers bars. I also love Kit Kats. Twix bars are pretty good too. Hot take, I do not like, what are they called? 100 grand bars? Yuck. No, thank you. Do not like those. But anyway, your favorite candy bar, whatever you guys like. That first one's delicious. It's awesome, hits the spot. And then you're thinking, oh, maybe I'll get another one, right? So let's say, for the sake of it, you treat yourself. You've got another one. You're like, okay, that one was not as good as the first one. But let's just say you're having a real bad day, right? Your girlfriend, your boyfriend, whoever broke up with you. It's time to binge some candy and watch your favorite Netflix shows. So you go for that third one. Now you're starting to feel a little bit sick, right? Because you only needed one, but you had 2 more. This idea of diminishing marginal returns, where the more we consume of something, the less favorable it is on the system in which it's consumed. It's an economics term, but it works out pretty well here. Hemoglobin is the same way. Hemoglobin starting out in the lungs is like, okay, sick. Like, I've got all this oxygen. It's delicious. I'm going to bind it up. Cool. We're going to shove that onto one heme. All right, cool. We've got some more oxygen. You mean I can bind 4? I can bind 3 more? Cool. I'm going to bind another one. As it binds 2, and then even as it starts to bind a third one, the hemoglobin molecule is like, I don't really need that much oxygen anymore. So oftentimes hemoglobin will stop binding oxygen molecules at right around 3 per molecule of hemoglobin. We have lots of hemoglobin per red blood cell. I don't know what the actual percentage of content is, but it's a lot. It's not like each red blood cell contains only 1 hemoglobin protein. We've got lots of it in there. So over time, as hemoglobin binds more and more oxygen, it starts to lose interest in that, right? So it's like, okay, I've got all this oxygen on here. We can kind of switch up our analogy and let's say, okay, hemoglobin's dating oxygen for a while, right? It's dating it for around 6 to 7 months, right when we got that third oxygen molecule. But then it starts to get that 6th to 7th month itch. You know what I'm talking about? Where you're dating somebody, you just get that ick, you don't know what it is, but you're just sick of the person, right? And you just gotta dip, you just gotta head out, right? So that's what happens to hemoglobin. It starts to lose interest in oxygen. And then right around this time kind of works out, oh, hey, it's walking along. It sees this other tissue. What's this tissue got? It's super saturated in carbon dioxide. Hemoglobin's never seen this before. It's new. It's interesting. It's alluring. It's like, all right, get rid of all this oxygen. I don't need this anymore. Let's nab up some carbon dioxide, right? But then the same thing happens as hemoglobin is like, oh, I've got all this carbon dioxide and, I really miss oxygen. Okay. Maybe I want oxygen back. Maybe I want to get rid of this carbon dioxide. This is a mistake. I want to go back to oxygen. So again, conveniently, right around this time, hemoglobin has circulated back into the lungs. And guess what's in super high concentration in the lungs? Usually oxygen, right? So hemoglobin has this perpetual self-destructive cycle of constantly going back to that, they go back to that ex who's really good to them, then they get bored, and then they dump them for the same person, and it's just this perpetual cycle, right? So hemoglobin's never going to learn its lesson, which works out coincidentally very well for us, because without it, then our tissues wouldn't get oxygen and we wouldn't be able to get rid of carbon dioxide. All right, silly analogies aside, we've got a little scanning electron micrograph. Oops, I didn't want to do that. We've got this, scanning electron micrograph of red blood cells floating through a vessel here. So we can see even in the wall of the vessel, we have those little pores I was mentioning. Those are the fenestrations that can enlarge in an inflammatory response. And then each of these things like neutrophils and other things that are floating around can migrate through those pores to, be part of an inflammatory response and fight off infections. But anyway, we can see that we've got red blood cells floating around. Some of them are misshapen, some of them are malformed. So these ones are, chances are, they're probably getting ready to be broken down and recycled and converted back into their components so that we can use them for producing new red blood cells. So as we just described, hemoglobin has various stages of affinity for certain gases, but the one thing that hemoglobin cannot resist is carbon monoxide. So not carbon dioxide, but carbon monoxide. In other words, the poisonous exhaust fumes that come out of our cars, right? So if you end up inhaling carbon monoxide, that carbon monoxide will stay in your system for an incredibly long period of time, because the factor of affinity for carbon monoxide to hemoglobin is over 100 times greater than it is for oxygen or carbon dioxide. So it stays bound to it very, very tightly. So part of the problem is that the more hemoglobin that gets exposed to carbon monoxide, then the less hemoglobin you have to bind up oxygen and carbon dioxide, right? So if that's the case, hemoglobin sticks and stays bound to carbon monoxide, and then it's unable to associate with other gases. So then our tissues become rich in carbon dioxide, called hypercapnia, and then the rest of our tissues also become deficient in oxygen, called hypoxia. So we develop these two conditions of too much carbon dioxide, not enough oxygen, and then our tissues will suffer. And then as a result, our tissues will undergo what's called an infarction, or they lose their supply of oxygen, and then they senesce and die, right? It's the same idea that goes into, a clot or an obstruction that reduces blood flow to the heart, resulting in a myocardial infarction, or specifically a heart attack. It's the same idea. So then we have an enzyme that will control our chemical environment of our blood as well. I realize it's kind of an abrupt transition there to talk about, carbon monoxide and, heart attacks, and then talk about enzymes that regulate our chemical environment, but it's still in the same ballpark of importance. If our chemical environment of our bloodstream is not appropriate, then none of our hemoglobin is going to work. They're not going to be able to bind oxygen or carbon dioxide. We won't be able to transport, hormones or enzymes or other things won't be able to operate effectively within our bloodstream. So we have this enzyme called carbonic anhydrase, which helps convert extra carbon dioxide in the presence of water into bicarbonate ions. So this is really nice if we develop too much hydroxide ions in our blood. So if our blood gets a little too alkaline, then we can convert that extra carbon dioxide into some extra hydrogen ions to bring our pH back down to where it's supposed to be. So our blood pH is generally anywhere from about 7.3 to 7.4 pH. So it's slightly alkaline, but it's fairly close to being chemically neutral. You can change that drastically with some of the foods that we eat, but again, that's the beauty of this carbonic anhydrase enzyme, where it can not only do the forward reaction, but we can also convert it and reduce or do the reverse reaction, where we can convert bicarbonate ion into carbon dioxide and water, and we'll release some hydroxide ions as well. So It's pretty cool. We can do the forward and reverse reactions, and carbonic anhydrase basically just sits there and like kind of flips a little switch every time it goes out of whack in one way or another. But let's say that you consume a really, really acidic food. Like you think it's a great idea to go out and drink a bottle of lemon juice, which do not do this, by the way. You will go to the hospital. Okay. So if you consume a lot of really acidic foods in a short period of time, you're at danger of undergoing a condition called acidosis, where your blood pH drops super, super low and carbonic anhydrase can't keep up with it. So bad things happen, enzymes stop working, proteins stop working. You get the idea. Chemical environment needs to be good, otherwise things don't operate. It would be like trying to run a marathon in sub-zero temperatures wearing only shorts and a t-shirt, right? Like you can't do it. You just can't do it. Okay. So what's kind of cool too is that our body can control how much red blood cells we have at any given point in time. So what'll happen is as we, let's say, we go up in elevation. If any of you guys like to, hike or like to, go up in the mountains or ski or snowboard or anything like that, what'll end up happening is as we increase in elevation, there's less pressure the higher up in the atmosphere we go. And so there's less oxygen available. So in order for our body to compensate for that, we need to increase red blood cell production so that we can extract as much oxygen from the atmosphere as we can. The kidneys produce this hormone called erythropoietin, so EPO. So this erythropoietin activates red bone marrow and causes an increase in the production of red blood cells, which will allow for us to extract more oxygen from the atmosphere. So literally what ends up happening is our blood gets thicker the more time we spend at elevation, or the more time we spend actually it's not necessarily colder temperatures, it's usually just elevation that affects things like erythropoietin activation. So part of the problem with that, though, is you can have too much of this occur. There's medical conditions that we'll talk about at the end of the class today. I forget what their names are, but you can have too much erythropoietin in your system at any point in time, and you can have too thick of blood. So if your hematocrit gets too high, basically you have too many red blood cells per unit plasma, then your blood becomes too thick, and then your heart has to work harder to force that blood through your vessels, right? Which is not good. Okay, so erythropoiesis, okay? And then we have hematopoiesis, so our breakdown and recycling of the components of our red blood cell. So humans, our red blood cell life cycle is roughly 115 to 120 days, so right around 4 months. At any given point in time, we're cycling through roughly 3 million red blood cells pretty much every second, right? But remember, having 30 to 36 trillion at any given time, 3 million is kind of a drop in the bucket, right? But still, as we're breaking them down and recycling them, we're also producing them at an equivalent rate. So what ends up happening is we have macrophages that are present within the spleen or the liver that essentially filter and sense when red blood cells need to get broken down and recycled. So if their membranes are torn and damaged, if they're small, if they've been, kind of shriveled up, if they just look like they've been through the ringer, macrophages in the liver and spleen say, all right, it's time to go to the farm. You need to spend the rest of your days on the farm. So then macrophages chomp them, and then they break them down and they break them up into their components, right? So one of the main things that we're interested in recycling is this hemoglobin protein, okay? So what we end up doing is we kind of recycle and we preserve that heme, we extract that iron ion, and we can even break down the protein itself into its constituent amino acids, and then reuse those to rebuild and refold each of the globin tetramers of that protein. So this heme ends up going through a couple different manufacturing processes until it eventually reaches the stage that's called bilirubin. So bilirubin is preserved as a kind of yellowish pigmented liquid that's stored in the liver and then released into the blood and then eventually into the intestines where it's released into the bile. So bile will then end up getting extracted out of our intestines via the excretory system, and then we can release that waste product through our kidneys as part of our nitrogenous waste in our urine. But if you have problems or you're unable to cycle through the bilirubin, it can build up in the liver and cause a condition called jaundice, which will inflame the liver and impair its function. This will usually manifest as, a yellowing complexion in an individual, and usually kind of like feeling chronic fatigue. So a common instance of this occurring, it's not really, I should say it's a common mix-up that can happen in early childhood. Lots of baby food is really rich in things like carrots and squash, which have a pigment called carotene in it. You probably know where this is going because I've given you guys the same example before. But just in case you forgot, what'll happen is the baby's skin will turn yellow or yellowish orange. And correctly, parents will get worried and take their kid to the doctor. And the doctor's like, okay, what are you feeding him? Lots of carrots and squash. All right, cool. Stop feeding him that because he's probably eating too much, and you might want to tone back on that, and then his skin will go back to whatever color it is, right? So bilirubin is a yellowish pigment, whereas carrots and squash and those kinds of things have an orangish pigment. But anyway, they can look similar. So jaundice, again, is that condition where the liver is unable to cycle through bilirubin. Okay. Let's see. So here we've got a light micrograph of, human blood smear. We can see we've got some neutrophils and monocytes, lymphocytes that are present here. We can see some platelets off over here. Then all of our little red blood cells, no nuclei in them. Pretty interesting. So I've got this little blurb here on heart attack treatment. We'll talk about this more in our next chapter as well. So one thing that's very important, obviously, for clotting is platelet activation. And we can use these chemicals that platelets contain called thromboxanes to activate other platelets, right? So that's what they release. So part of the problem with some people that have heart attacks come about for a variety of different reasons. There's myocardial thromboses where you develop clots, which are different from plaque buildup. But if you have overactive platelets, you can develop little clots in your blood, and those clots can travel from one part of your circulatory system to the other. So sometimes what will happen is if these clots are known by a physician or a cardiologist, sometimes patients will get prescribed with aspirin, which can help inhibit thromboxane activity. So it basically stops the signaling between platelets so that clots won't form. But part of the problem with that is if those people get injured, if they get a cut or something, the chances of them losing a lot of blood before a clot can form are incredibly high. All right. We also have some other medications called tissue plasminogen activators, or TPAs. So those will help inhibit the activity of clot formation. So they basically prevent fibrinogen from going and searching out these sites and creating clots as well. Right. This is obviously, a big issue for women who are pregnant too, who are at risk for clots forming or things like eclampsia, where, sometimes aspirin might be administered to help reduce things like clot formation, but it can be a really big issue and needs to be taken with extreme caution, especially like leading up to labor and delivery, because if there's hemorrhaging, odds of bleeding out are incredibly high too. Okay, I'm going to skip over some of these pictures here. I mean, neutrophil, basophil, eosinophil. Okay, so that orangish reddish complexion. Then lymphocytes, monocytes transforming into macrophages. All right. Okay, so let's look at how a clot forms. So what's kind of cool about how a blood clot will form in a damaged vessel, right? So kind of some perspective here. So this is our wall of the blood vessel, and this is the inside of our blood vessel. We know that because we have endothelial cells that are present here, right? So endo meaning within. Okay. So at the site of an injury to a vessel wall, okay, what will end up happening is these little proteins that are exposed on the surface of this wall, called von Willebrand factor, act as essentially beacons for platelets to go and bind to. So when that happens, when von Willebrand factor is adhered to by a platelet, the platelets will release these chemicals that they contain in them called thromboxane, and they'll also release some ADP. ADP will act as a secondary signaling molecule in conjunction with thromboxane to activate other platelets that are floating by as an apocrine response to be able to say, hey, come on over, we need to block up the site and prevent further damage to this area. So then platelets will then start to aggregate around different fibrinogen receptors. So these little red proteins that are on the surface of each of these platelets will bind to fibrinogen. And remember, fibrinogen is one of those immunoglobulin proteins that's floating around in the blood. Those fibrinogen proteins will then bind to exposed fibrinogen receptors and then allow for the adherence of other platelet cells, or cell fragments rather, to start sticking together. And so you can see that as the more and more of these start to bind up, the more thromboxanes start to get released. You can see how this positive feedback loop can start to create a really large scale response, and we can get a lot of platelets being bound very, very quickly. One of the other cool things that blood vessels will do is they will undergo what's called vascular spasming, where they can also release thromboxanes to trigger the attraction of other platelets that can be in the area. So this spasming, or rapid contraction and relaxation, will cause the release of those thromboxanes. That's kind of cool. Sometimes people can be deficient in von Willebrand factors, so they aren't able to produce this protein. And so you can get injections or have medication that contains von Willebrand factor to help with any issues that you might have with clotting. All right. So here's our basic diagram that kind of shows how a clot gets formed. This is very simplified. So if we have injury to a blood vessel, that connective tissue gets exposed. We release things like thromboxane via vascular spasming. This will activate inactive clotting factors, so it'll turn them on basically. And then those active clotting factors will then utilize things like calcium and other platelet chemicals like ADP to then activate prothrombinase. Prothrombinase will then convert prothrombin into thrombin, which then converts fibrinogen into fibrin. So fibrin is the actual clot, right? But fibrinogen acts as that initial protein receptor. So we've got this whole kind of cascade sequence in order to create our actual clot. But usually what ends up happening with people who end up having issues with clotting is there's a disease called hemophilia, which is a sex-linked disease that only males can get because you have to have, it's carried on the X chromosome, but if you are a female, you can't actually express hemophilia because because you don't have the Y chromosome. So males that only have one X chromosome will express the hemophilia disease if they contract it, or if it's passed down through the genome. There are instances of males being carriers for the disease, but it's pretty rare. Usually, if the mother is a carrier for it, if she has a son, chances are the son will get hemophilia. Most people with hemophilia lack an important enzyme that converts fibrinogen into fibrin. There's like 15 different enzymes that are responsible for that. And there's some specific ones that they're responsible, or that they lack. One of my students at the high school that I teach at has hemophilia, and I forget which one it is. So there's a couple different types of hemophilia and all this stuff, but yeah. So this process is a little bit more involved. Okay, so now the clot has formed. We've created our fibrin plug. Now, all right, the tissue's been repaired. We need to get rid of this clot and break it down. Okay, so we use tissue plasminogen activator, which will then convert plasminogen into plasmin, which then breaks down fibrin into its components, right? So we can release fibrinogen and then get all of our stuff back and kind of recycle that. Okay, so TPAs, or tissue plasminogen activator medication, will help break down clots for this reason. Okay, questions before we move on to blood typing? Give me a thumbs up if you guys are good. All right, cool. Thanks, guys. All right. So blood clots, or blood types rather. Blood types typically fall into 4 different categories, and these are those categories. There's actually a 5th one that I learned about recently. It's super, super rare. It's called Rh null blood type. And so that's where they don't have any antigens. They also don't produce any antibodies either. It's a pretty rare condition, so they are like a universal donor and technically universal receptor as well, or receiver of blood transfusions, which is pretty nuts. I think it's something like less than 4% of people in the world have it. It's pretty crazy. And there's what's interesting too is that there's all different kinds of demographic data that show that there's certain percentages of blood types that exist for people who are Native Americans versus Native South Americans versus Latin America. Wherever it ends up being, there's all these kinds of interesting demographics that show that there's consistent blood typing within different parts of the world. South America, I think there's a universal blood type for most people that are Native South American. Forget what it is. But in America, in North America, the most common blood type is actually type AB. I believe it's AB positive is the most common blood type. So people who are type AB contain both sets of what we call surface antigens, which are basically chemical identifiers that the immune system registers. It's like, okay, these are what we have. So if we have one, then we need to produce antibodies that fight against the other, vice versa. But people who have both surface antigens don't produce any antibodies. So the cool thing about type AB individuals is that they can receive any kind of blood type as long as they either have the Rh factor, or if they don't have Rh factor and they haven't been sensitized to it, then they can still receive a transfusion that way. But if they have been sensitized and they produce the anti-Rh antibodies, then they can only have type A, B, or AB negative blood, or type O negative. All right. So pretty much the way that we identify different blood types is we have to figure out what the kind of surface antigen is. So each one of these little surface antigens, like I said, is the chemical identifier for what kind of blood we have. So antigen A looks one way. So people who are type A blood, I think type A is actually the second most common in our country, which is I think it's like 40-something% of people have type A. So I have type A positive. I don't know if any of you guys know what blood type you have, but if you have type B, you're in one of the rarer categories. So type B negative, I think, is one of the most rare in our country. But so type A and type B will produce the corresponding antibodies that will fight against that, right? So if you have type A blood, like myself, then you produce anti-B antibodies. So if you get a transfusion with somebody who has type B blood, then your body, your immune system will attack those type B red blood cells, causing something called an agglutination reaction, right? Same thing goes for somebody with type B. They'll fight against any kind of type A blood. But type O individuals are kind of interesting. They're the universal donor, but they can only receive blood from other type O individuals because they produce both sets of antibodies that'll fight against both A and B surface antigens. All right. So the way that works is that, if you get a transfusion or a donor of blood with the same type, then the antibodies that you produce won't attack those red blood cells. So everything's cool. But if you receive a blood transfusion of somebody who has the corresponding blood type that fits the antibodies you produce, then those agglutination responses will occur because those antibodies label those red blood cells for destruction by things like the neutrophils or T cells. So sometimes, though, it can be a little bit more interesting, especially with pregnancy. So with Rh factor, which is this second set of identifiers that our red blood cells will produce, it's also called the D antigen, but clinically it's you can also call it the Rh factor. So there's a condition called hemolytic disease of the newborn, where sometimes if the mother is Rh negative, so she doesn't have the surface antigen for this Rh factor, if she hasn't been sensitized to Rh factor, then her body won't produce antibodies that will fight against Rh factor. So if she's pregnant and is carrying a child that does have Rh factor, what will end up happening with the first pregnancy is that usually like hemorrhaging during delivery, or sometimes there's a tear during development between the placenta and the maternal blood supply, then the mother's immune system will become sensitized to that Rh factor and will start producing antibodies. Usually, the fetus that is the first one that goes through this developmental stage isn't affected by the mother's immune system because the mother's immune system hasn't been sensitized long enough. But usually, the second pregnancy, if the fetus is Rh positive, the mother's immune system will start to attack the red blood cells of the developing fetus, developing a condition called hemolytic disease of the newborn, where the newborn's red blood cells will all end up getting destroyed by the mother's immune system. This is treatable now, and we can utilize things like immunosuppressants, or in extreme situations, we can actually do fetal blood transfusions in worst case scenarios. But it can be treated, a little bit more effectively nowadays, because it used to be, an untreatable occurrence before we knew what was going on. All right, so let's round this out with talking a little bit about some various kinds of diseases that can affect the blood. So here, this figure shows you guys a centrifuge machine that measures what's called hematocrit. Like I mentioned before, hematocrit is basically the percentage of red blood cells relative to how much plasma you have. So there's a sexual dimorphism where males tend to have anywhere from 5 to 10% greater hematocrit than females do. So some of that, can be for a variety of different reasons. Sometimes it's in, it's a disease condition known as anemia, right? So anemia is a generalized term that represents a deficiency in normal hemoglobin content, right? So this results in decreased red blood cell production. If you don't have as much hemoglobin, then you don't produce as many red blood cells. There's a whole bunch of different kinds of anemia that can be developed. So aplastic anemia is the inability just to produce red blood cells flat out from red bone marrow. So you rely mostly on things like the spleen and the liver to do that. You can have iron deficiency anemia, where due to dietary deficiencies or any other kind of issue, you can develop a hemoglobin deficiency there. One thing that's really interesting too is important vitamins for the production of hemoglobin involve things like our B vitamins, specifically B12. So pernicious anemia is a condition where you have a B12 deficiency in the diet. What's crazy about this is fairly common in people that live in northern latitudes, people who aren't exposed to sunlight very frequently. So lots of people in England back in like the early 1900s would suffer from pernicious anemia, and they would die because we didn't know that it was a simple condition of supplementing their diet with B12 vitamins, and it would actually treat the issue. So kind of interesting. Hemorrhaging anemia due to loss of blood. So things like heavy flow during the menstrual cycle can contribute to things like hemorrhagic anemia. You have hemolytic anemia, where you can have an where recycling and breakdown of red blood cells is overactive. So hematopoiesis frequency is elevated. And then you can have thalassemic anemia or thalassemia, where you have a genetic disorder for a lower hemoglobin production. So there's no real necessarily cure for that, even though that's not strictly true nowadays with the advancement of technology, things like CRISPR, where you can actually edit the genome and delete sequences for genetic disorders for things like that. There's lots of moral and ethical issues that go into that, and the technology is incredibly accurate, but it's not really available for mainstream use of things like that. And then obviously we have sickle cell anemia, where due to a reading frame mutation, where you end up deleting a single nucleotide sequence in the polypeptide chain to make our protein. Pamela, give me a second, and I'll answer your question. With sickle cell anemia, if you lose that nucleotide pair, the whole reading frame for the translation shifts over, and then the protein ends up being completely different than what it's supposed to look like. So sickle cell anemia is another genetic condition that can result from that. interestingly enough, I think I'd already mentioned this. I don't know if I did, but sickle cell anemia is a adaptation to fight against diseases like malaria, which can attack red blood cells too. Yeah, Pamela, you had a question.
So I suffer from anemia
Mhm.
deficiency, but it's to a point where I've been like once previously I was diagnosed with first stage of leukemia because my anemia dropped so low.
Mhm.
So, but I didn't quite understand why, and it seems like I'm starting to go through that again. Almost 10 years later, I'm having to go through almost the same process again because my body doesn't seem what I'm being told it's not producing a specific type of blood cell, or my body's producing it more than it should.
Hmm. So is it a hormonal thing like erythropoietin?
What That's what I thought it was, but you explaining it right now, the hemorrhaging one,
Uh-huh.
um I suffer from that, and
Mhm.
but but they haven't diagnosed me with that. They have just said that basically I produce more of one or the other blood cell that is causing my anemia to drop so low. But that can happen where the anemia falls too low and you produce a cancer cell.
That is a good question. I don't actually know. Um, if you, I mean, it sounds. I mean, if your physician's diagnosed you with that, then probably. But that's, to be honest, I wasn't actually sure about that. So that's a good question, though. Um, yeah, I mean, definitely, ask for more clarification. The next time you're with your physician and kind of maybe have them try and explain to you a little bit more about what's going on because you know again, I'm not a physician or anything like that. I'm not a practicing clinician or anything like that, but it sounds like that might be what they're saying is if you have something like hemorrhagic anemia or hemolytic anemia where you're breaking down too many of your red blood cells, but like what you were saying, you said that your physicians are telling you that you're overproducing a certain kind of white blood cell. Is that what it is? Because if
Correct.
you're, because okay, because yeah. So I don't. Yeah, I wonder which one that would be. Maybe it's neutrophils, and so maybe you just have like overactive macrophages.
I don't know. It's just the way you were explaining it kind of makes sense of what my body was kind of doing. So that's why I was like, I've never been clear if that really can cancer cell can be produced based off of very extremely low levels of anemia.
That's interesting. So yeah, I wonder, or well, I wonder if it's either it's probably either neutrophil or monocyte activity being incredibly heightened, which would result in your, I guess the way to phrase that way would be your anemia levels would be elevated, even though I mean, so you would have a greater instance of anemia. Your anemia would be more pronounced at that point. It sounds like, but yeah, that's really interesting. I mean,
Okay, thank you. I just want a little bit more clarification on that, and I wanted to see if you knew anything about it. So I appreciate it.
yeah, I mean, well, thank you for sharing that. I appreciate you doing that. So, I'm sorry I can't be more help, but um, you know, you can always send me an email or do some research too, and then let me know what you find. Be interesting. Okay, so let's round this out with, I'll just kind of put this up here so you guys can peruse over some of these representative diseases and disorders of the blood. So I've already mentioned things like hemophilia and von Willebrand disease, malaria, septicemia. So this is what can come about through sepsis or things like fecal matter getting into our circulatory system can cause a widespread inflammatory response that drops blood pressure and puts the body into shock. And so then your tissues get oxygen deprived. then you can have different kinds of erythrocytosis where you can overproduce red blood cells. That's what I was mentioning before, where you can overproduce too many red blood cells and then your hematocrit levels go way, way up. So you don't have enough plasma, you don't have enough um, uh, immunoglobulins, albumins, fibrinogen. You don't have enough of that in your blood. The blood becomes too thick, and then the heart has to overwork itself. Yeah. So hypertension, that's the word I'm looking for. And then obviously things like acquired immunodeficiency syndrome, AIDS, or the advanced stage of human immunodeficiency virus, or HIV, which can affect lymphocytes and suppress the immune system. So you guys are probably already aware of this too, but nobody dies from AIDS or from HIV. They die from diseases that are normally, insignificant, that would be no problem for most people with healthy immune systems, but since their body essentially has no immune system, they end up, succumbing to a common cold that gets upgraded to walking pneumonia and then just slowly deteriorates their body. Yeah. All right, guys, that is it for today. Does anybody have any questions? We're not, I mean, it's 6:07. We've only got like 13 minutes left, so I won't burden you with talking about the heart yet. We'll save that for break. But I would encourage you guys to spend some time reading through chapter 12, familiarizing yourself with some of those concepts. Chapter 12 is again, one of our more physiologically demanding chapters because there's just a lot of cool stuff that goes on with the heart. Let me stop our recording here. So we'll